In this example, we're going to use NAVA to visualize the experimentally determined secondary structure of an entire HIV-1 RNA genome. Specifically, we're going to be looking at the NL43 HIV species. The secondary structure of this genome was published in a landmark paper back in 2009, where the authors used an experimental technique called SHAPE to determine the secondary structure of this sequence. Simply explained, SHAPE is an experimental methodology which uses chemical probing to measure chemical accessibility at each nucleotide position in a particular sequence. Nucleotides which are unpaired in the sequence tend to be more accessible to the SHAPE chemistry, whereas nucleotides which occur in paired regions in the sequence tend to be less accessible to the SHAPE chemistry. Using this measure of SHAPE accessibility at each nucleotide position in the sequence, the authors were able to gather information which helped them constrain a thermodynamic folding algorithm, which then allowed them to very accurately determine the secondary structure of this genome. We are now going to visualize this experimentally determined secondary structure using NAVA. The first thing that we're going to do is open NAVA. Upon doing so, NAVA requests that we either start a new project or that we open an existing project. We're going to start a new project and we're going to call it Example Project. In order to begin an analysis, we're going to import 5 files that are related to the NL43 secondary structure. The first of these 5 files corresponds to the experimentally determined NL43 secondary structure itself. This file just contains information about which nucleotide positions are base paired with which other nucleotide positions in the sequence, and which nucleotide positions are unpaired. The second file is a spreadsheet file containing shape reactivities. If we open the spreadsheet in Excel, you will see that there are 4 columns. The first of these 4 columns gives the nucleotide position in the sequence. The second column gives the sequence at each nucleotide position. The third column contains shape reactivities, and this is going to be the column that we're most interested in for this example. And the fourth column contains pairing probabilities, but we won't be considering this column in this example. The third file that we have is a file containing the NL43 sequence, and this is obviously the sequence that corresponds to the NL43 secondary structure. The fourth file is a GenBank file that contains sequence annotations, and these sequence annotations give the locations of genes and other features in the NL43 sequence. Finally, we have a file containing 50 HIV-1 subtype B sequences. NL43 is itself an HIV-1 subtype B sequence, and we're going to be using these 50 sequences to overlay a slightly different type of information on the NL43 secondary structure. We're going to import these 5 files by selecting them and dragging them into the project navigation panel in NAVA. Once NAVA has completed importing these 5 data files, they will appear in the project navigation panel on the left-hand side of the application. Every time NAVA imports a data file, it will automatically detect the file format of that file and categorize it into one of 6 data categories. Data files that have been imported into NAVA are no longer considered data files, but are rather considered data sources, because NAVA uses its own internal representation to represent those files. If we click on one of these data sources, NAVA will give us some information about that data source, some of which is editable. In this example, we've selected the NL43 sequence, and NAVA has categorized this data source as an alignment data source. In particular, it's a nucleotide alignment. It consists of just one sequence, and that sequence has 9,173 positions, which is the typical length of an HIV-1 genome. If we select the 50 HIV-1 subtype B sequences that were imported, once again, this is an alignment data source. It's a nucleotide alignment data source. This time consisting of 50 sequences, as we already know. But these 50 sequences consist of only 2,769 column positions, which is very much shorter than the NL43 sequence. Now, the reason for this is because this is just corresponding to a truncated region of the HIV-1 genome, in particular a region that we're going to want to investigate later in this example. You can see here that the NL43 annotations have been imported, as well as the secondary structure, which is important, and finally the spreadsheet containing the shaper activities. Now if I click on this arrow over here, it will show me the columns in that spreadsheet, and as you can see, it's the 4 column positions that we saw earlier. Now we want to visualize the NL43 shape structure, and in order to do so, we click on the Visualization tab. In order to visualize a particular secondary structure, it first needs to be added as a data overlay. So I'm going to go to Add Data Overlay, Add Structure, and this will bring up a dialog that allows me to specify some parameters about the secondary structure I want to visualize. Firstly, I'm asked to select the secondary structure data source that I want to use. In this case, there's only one data source available because we've only imported one secondary structure into the current project. So I'm going to select the NL43 shape secondary structure. Next, I'm asked to specify the conformation of the sequence corresponding to the structure. In this case, it's a linear sequence, but in some cases the sequence can be circular, which means that the 5' and 3' ends of the sequence are covalently linked. However, in most cases, the sequence is going to be linear. The next thing I'm asked to do is to choose a mapping source. A mapping source is an important piece of information that informs NAVA of how to map other data overlays to a particular structure overlay. A mapping source is a sequence or an alignment of sequences where the positions in that sequence or alignment of sequences correspond exactly to positions in the secondary structure data source. In this case, the mapping source is trivial. It is just the NL43 sequence itself, which does correspond to the NL43 shape secondary structure. Finally, I'm asked whether I would like to add this mapping source as a nucleotide overlay, and I do want to do this, so I'm going to keep this box checked. I'm then given a warning which tells me that the length of this mapping source does not match the length of the structure. It just so happens in this particular example that this secondary structure is slightly truncated, but I do know that the first 9,170 nucleotides of this secondary structure do correspond to the first 9,170 nucleotides of the mapping source, so this isn't a problem. Lastly, I'm asked to create a list of substructures. NAVA will automatically have generated a list, but I'm able to edit that list by selecting Edit List. This will bring up a dialog where I'm able to specify a number of parameters that will change the generated list. NAVA requires a list of substructures because it cannot display the entire secondary structure which we've specified in one go. Instead, it subdivides up that secondary structure into a number of smaller substructures which can easily be visualized. I'm not going to make any changes to this list, so I'm going to click Cancel, and then I'm going to add the structure overlay. Once the structure overlay has been added, the first substructure corresponding to that overlay will appear in the center panel of the Visualization tab. If we look to the left of the center panel, we will see a list of 4 directories corresponding to the 4 different types of overlay that NAVA supports. If we look in the Structures directory, we will see an item corresponding to the current secondary structure overlay that is being viewed. Next to this item is an eye icon indicating that this overlay is being visualized. If we look in the Nucleotide Overlays directory, we will see an item which corresponds to the nucleotide overlay, which is automatically added when we added the structure overlay. We can view this item by right-clicking and selecting View. This will now overlay the NL43 sequence on top of the secondary structure overlay. We are now going to add some annotations to the Sequence Annotations panel. This can be done by right-clicking and selecting Add from Source. For this particular project, we've only imported one set of annotations, and so only one set of annotations appears over here. These are the annotations we want to add, so we're going to go ahead and click Add. Doing so will automatically start mapping those annotations to the currently selected structure. The annotations will appear in the Sequence Annotations panel. You will notice that some of the annotations that have been added are slightly irrelevant, so I'm going to remove them. I'm also going to rearrange these annotations for better visualization. Right-clicking and clicking stack annotations will automatically do so. The annotations can also be shifted up and down by right-clicking on an annotation and clicking Move Up or Move Down. Further annotations can be removed by right-clicking, selecting Remove, and choosing the annotation you want to remove. The Sequence Annotations panel allows us to easily navigate through substructures corresponding to the current structure overlay. The position of the currently selected substructure is indicated by a green block. By mousing over the Sequence Annotations panel, the position of other substructures is indicated using blue blocks. By clicking at a particular position, the corresponding substructure is opened up in the viewer. For this particular example, I'm interested in a substructure that occurs in the envelope gene of the HIV genome. By mousing over to that position and clicking, I can open up that substructure. This substructure is the rev response element. The next thing we're going to do is overlay the shape reactivities on the structure. In order to do this, we're going to go to Add Data Overlay, and we're going to add a one-dimensional overlay. This will bring up a dialog that allows us to specify some parameters about the overlay we want to add. In step 1, we are asked to specify a data table, and in this project there's only one data table. It is the table containing the shape reactivities. From that table, we are asked to specify a data column, and in this case we're going to choose the shape reactivity column. In step 2, we are asked to specify how values in this table are positioned. In this case, the values are positioned naturally. In other words, the numbering starts at 1, and the first line in this table is a header. If we look at the data preview panel on the right, we can get a preview of how these values are positioned, and I can see here that the values are positioned correctly. So this means I don't need to make any changes at step 2. In step 3, we are asked to map the values to the structure using a mapping source, and in this case the mapping source is once again the NL43 sequence. Positions in this NL43 sequence correspond exactly to positions in this table. In step 4, we are asked to specify the numeric range of values we want to visualize. In this case, I'm going to leave the minimum value as 0, but I'm going to set the maximum value to 1. Then we are asked how we should treat values out of this range. We can either treat them as missing data, or we can treat them as clamped. If we treat values out of the range as missing data, this means that they will be excluded. If we treat values as clamped, this means that if they are out of the range, they will either be treated as the maximum value if they are above the maximum value, or they will be treated as the minimum value if they are below the minimum value. In this case, I'm going to leave it as clamped. In step 5, we are asked to choose how we wish to display the data values. On the left-hand side, you will see a color legend which represents how each numeric value will be depicted using a color value. To the right of this legend, you will see a histogram which represents the distribution of values. We can change the color legend by double-clicking on the color tab to change a color, and we can add a new color by left-clicking to the right of the legend. What I'm going to try to do here is to replicate the color legend that was used in the paper that experimentally determined the secondary structure of this NL43 sequence. In this paper, the color legend that they used was not a color gradient, but rather a set of solid colors that they used to represent their different numeric values. Now that I've done that, in the data preview panel you can see a preview of what each numeric value will look like in terms of a color. I'm now going to add this as a one-dimensional overlay by clicking Add. And once the one-dimensional overlay has been added, we can view it by right-clicking on it and selecting View. And now you'll notice that each nucleotide within the structure has been assigned a color. In particular, nucleotides with a low shape reactivity are colored in black and green, and nucleotides with higher shape reactivities are colored in yellow and red. If you look closely at the structure, you will notice that the nucleotides that occur in the base paired regions of this molecule, in other words the stem regions, they tend to have low shape reactivities, whereas the nucleotides that occur in the loop regions, the single-stranded regions, those nucleotides tend to have higher shape reactivities. And this is exactly what we would expect, because if you remember correctly, the nucleotides with a low shape reactivity are the ones that are inaccessible to the chemistry, whereas nucleotides with a high shape reactivity are those nucleotides that are accessible to the chemical modification. And this is how the secondary structure of this genome was determined. One last thing to look at over here is this color bar that runs along the length of the genome, and this gives you an indication of the shape reactivities at different positions along the genome. If you look at this position over here, which is the position of the substructure that we're currently viewing, you can see that it has a particularly low set of or distribution of shape reactivities occurring in this region. The last type of data overlay we haven't spoken about yet are two-dimensional overlays. In order to add a two-dimensional overlay, we need to have a matrix data source available. In the current project, we do not have a matrix data source. However, we can generate one by going to the Data Input tab. In the Data Input tab, we have a list of currently available data sources. By selecting an alignment data source, in this case, a set of 50 HIV1 subtype B sequences, and using the ppfold action, we can generate a base pairing probability matrix by performing a ppfold secondary structure prediction. In order to do this, I select both the alignment source and the ppfold action and click Run. When I do this, however, I get a warning that informs me that the selected alignment source may be unaligned. In order to perform a secondary structure prediction, ppfold requires an aligned set of sequences. So I'm not going to run ppfold just yet. Rather, I'm going to use one of the 3 built-in alignment tools to align these sequences. In this example, I'm going to choose to use the mapped alignment tool. When clicking Run, I get a settings dialog which allows me to specify how I want to align these sequences using the mapped tool. I'm just going to use the automatic setting to align these sequences. Clicking Run will begin aligning these sequences. Once the mapped application is completed running, the aligned data source will appear in the project navigation panel on the left. Now that we have an aligned data source, we can use the ppfold application to perform a secondary structure prediction. Once the ppfold application has completed running, the predicted secondary structure and base pairing probability matrix will appear in the project navigation panel on the left. Now that we have a base pairing probability matrix, we can visualize it using a two-dimensional data overlay. To do this, we're going to go to the Visualization tab, we're going to go Add Data Overlay, and we're going to choose to add a two-dimensional overlay. In step 1, we are asked to select the data matrix that we want to visualize. Entries in this matrix represent interactions between pairs of nucleotides. In this example, we only have one data matrix to choose from, which is the base pairing probability matrix that we've just generated, and so we're going to use that matrix. Entries in this matrix represent base pairing probabilities for pairs of nucleotides. We're going to give this overlay a title, and we're going to call it HIV subtype B pairing probabilities. In step 2, we are asked to specify how the values are positioned in this matrix. In this case, the numbering starts at 0, so we're going to use this option. In step 3, we are asked to specify which parts of the matrix we would like to use. In this example, This matrix happens to be symmetric, and so values are repeated on either side of the matrix. Because of this, we're only going to use the upper triangular portion of the matrix because we don't want to display repeated values. In step 4, we are asked to specify a mapping source. In this case, the mapping source is incorrect. We need to specify the HIV subtype B sequences that were aligned and used to generate this particular base pairing probability matrix. In step 5, we are asked to specify the numeric range of values we want to visualize. And over here, I'm going to set the minimum to 0.01. We also asked to specify how we want values out of this range to be treated. And I would like the values that are out of the range to be treated as missing data. In other words, they won't be displayed. Finally, we asked to specify a color gradient. Over here, I'm just going to use this white to red color gradient, where white represents pairs of nucleotides with a very low base pairing probability, and red represents pairs of nucleotides with a high base pairing probability. Now that we've done that, we're going to add this two-dimensional data overlay, and we're going to right-click on it to view it. Now you will notice that between certain pairs of nucleotides within the structure, we have these red lines, and these red lines indicate base pairing probabilities. By mousing over these red lines, a little tooltip will appear, giving us the value of the base pairing probability for that interaction. What's interesting about these computationally predicted base pairing probabilities is that many of the computationally predicted base pairing probabilities occur between nucleotides that are predicted to be base paired within the structure. However, some of these high base pairing probabilities occur between nucleotides which are not base paired with one another in the structure. And those are interesting because they either indicate false positive interactions or they indicate true interactions that happen to be alternative base pairings within the structure. Another thing that you can do with these two-dimensional overlays is you can limit the interactions by moving the slider. And what this will do is it will only show the interactions that occur close to one another in the structure. By varying the slider, you can vary the distance of the interactions that you want to display. The final thing I would like to do here is to change the nucleotide overlay to a different nucleotide overlay by adding a nucleotide overlay corresponding to the 50 aligned HIV1 subtype B sequences. I can do this by going to Add Data Overlay and selecting to add a nucleotide overlay, and then selecting the HIV subtype B sequences that were aligned as the nucleotide source. After I've added them, I can right-click on them and view them on the structure. By zooming into the structure now, you will notice that within certain nucleotides there are sequence logos. These sequence logos correspond to nucleotide composition at each position in the structure. This nucleotide composition corresponds to the alignment of 50 HIV1 subtype B sequences. Nucleotide positions with high degrees of variation are represented by sequence logos with many base characters, and nucleotides with low degrees of variation are represented by sequence logos with only a few base characters inside the sequence logo. The relative proportions of each base are represented by the height of each character. Hopefully what this example demonstrates to you is that NAVA can overlay different types of data corresponding to either computationally inferred data sources or experimentally determined data sources. And by doing so, we hope that NAVA can assist researchers in gaining insights into secondary structures that they are interested in. That's it for this video. I hope it's given you some understanding of how NAVA works and how it can give you biological insights into nucleic acid secondary structures you might be interested in. For more information and to download the NAVA application, which is freely available, you can visit the link in the description below.